patterns of inheritance. I think before I said that DNA was my favorite, but I lied, because it's actually patterns of inheritance. So in order to talk about patterns of inheritance, we cannot talk about this without mentioning Gregor Mendel and Mendelian genetics. So Gregor Mendel was a monk who bred peas and discovered a lot of the information about how traits are passed from parents to offspring. And he did this by looking at peas. So one of his experiments was to breed true breeding yellow peas, meaning every time you bred these peas, they were always yellow offspring, with true breeding green peas. Every time he bred the green peas, he always got green offspring. But he got some curious results. Every offspring of these two was a yellow pea. So he labeled the yellow as dominant. This trait was dominant because every time you bred a yellow to a green, you got all yellow offspring. For his next experiment, he crossbred these yellow offspring. And he got very curious results. Most of the offspring were yellow, except for a few, which turned out to be green. He calls this green trait recessive. We now know these traits as alleles, and these are the variation in the gene that you inherit from your parents. So, in these yellow, one variation is to have 2 copies of the yellow gene. One variation is to have 2 recessive copies, and you get a green pea. So, we call this homozygous, meaning same. So you have 2 copies of the same gene. So the yellow is homozygous dominant. And the green is homozygous recessive. As we saw with the offspring of the yellow and the green, you can also be heterozygous, meaning you have 1 copy of each. Now, up here, being heterozygous, homozygous, is called your genotype. What genes you actually received from your parents is the genotype. The trait that you see being yellow or being green is called the phenotype. What do we actually see? So we've developed something called the Punnett square in order to track how traits are passed from parents to offspring. So let me show you this with the yellow peas. So we bred the yellow peas, we, meaning Mendel, I wasn't there, yellow peas and the green peas. And we know that their genotypes were 2 homozygous dominant copies and small y's had a homozygous recessive. So I take these 2 and I put them over here. So put the big Y here, I put a big Y here. These guys I take and I put over here. And then we kind of follow this like a multiplication table. I always put the dominant gene first, so that's the big Y, and then the little y, a big Y and a little y, a big Y and a little y, and a big Y and a little y. So it's helpful to keep an inventory of how your genotypes match to your phenotypes. So 2 big Y's is yellow, because yellow is dominant. Being heterozygous also means you're yellow. And only when you're homozygous recessive do you get to be a green pea. So we see here that 100% of these offspring are heterozygous and therefore are yellow. But let's check this out if we do his second experiment, where he took the heterozygous yellow plants and bred them together. Okay, so he took heterozygous yellow and he bred it to a heterozygous yellow. So again, we put one parent here and the other parent over there, and then we just follow it out. So we have 2 big Y's, big Y and a little y, big Y and a little y, 2 small y's. So if we track out our genotypes and phenotypes, they had a 25% chance, so 1 in 4, of being homozygous dominant. They had a 50% chance of being heterozygous and a 25% chance of being homozygous recessive. For our phenotypes, we had a 75% chance of yellow and a 25% chance of green. So these are the phenotypes, what you see. Genotypes are the alleles you receive. So this is called complete dominance. So let's practice complete dominance together. In humans, tongue rolling is dominant to non-tongue rolling. So I always take my inventory of how the genotypes and phenotypes match up. So if you have homozygous dominant, you can roll your tongue. If you're heterozygous, you can roll your tongue. And if you're homozygous recessive, you cannot roll your tongue. What type of children result from a mother and father who are both heterozygous tongue rollers? So they're both these guys right here. So we're crossing the heterozygous and the heterozygous. So now we have to put them in our Punnett square. And now we just follow the chart. So what results? We have 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive, and as we saw with the P's, 75% tongue rolling, and 25% no in our phenotype. So be careful when you're completing these problems if the problem is asking you for the genotype of the offspring or the phenotype of the offspring. Because we have 3 genotypes here, but only 2 phenotypes because one of them is dominant. Complete the polydactyly example on your own, pause the video, and then come back and check your work. Okay, polydactyly, which is more than 5 fingers or toes, is dominant in humans. So it says use A, so 2 big A's means you have polydactyly. A big A and a little a means you have polydactyly. And having 2 small a's means you have the normal 5 fingers and 5 toes. Okay, so show the cross between a heterozygous polydactyly individual, so somebody who's heterozygous, and a normal 5-finger, 5-toed person, which is the recessive. So it doesn't matter which one you put where. I just like to put the first one on top and the second one on the side. And now we're going to follow this out. Okay, so when we cross a heterozygous individual and a homozygous recessive, we get 50% hetero, who are also polydactyly. So heterozygous is their genotype, polydactyly is their phenotype, and we get 50% normal, 5 fingers, 5 toes, homozygous recessive. Try one more, this one in mice. Okay, check your work. In mice, black fur is dominant, so 2 copies is black. Heterozygous, also black because black is dominant, and 2 small copies gives us gray. So we are mixing a heterozygous mouse with a homozygous recessive. Heterozygous mouse times homozygous recessive. So again, same result, 50% are black and heterozygous, 50% are homozygous recessive and gray. So just when you think you're getting the hang of this, I'm going to throw a wrench in here and introduce you to incomplete dominance. So this is an intermediate phenotype, so what we see is in between the dominant and recessive genes. So when you have homozygous dominant, you see red. When you are homozygous recessive, you see white. And if you are heterozygous, you're neither red nor white, you're pink. So let's try snapdragons first together. Petal color shows incomplete dominance. A red flower is dominant, so 2 R copies is red. 2 small R copies is white. And when you mix a big R and a little r, you get pink. So first, let's look at mixing a homozygous dominant and a homozygous recessive. So we get 100% of our offspring that are pink. 100% heterozygous, 100% pink. Try this one on your own, using the same genes, big R red, little r white, big R, little r pink. Okay, so this time you are crossing 2 heterozygous individuals. So, let's find out what we get. Okay, so we have a 25% chance of getting red, homozygous dominant, 50% chance of pink, heterozygous, and a 25% chance of white, homozygous recessive. Try one more. This one's an Andalusian fowl type of bird. All right, check your work. Okay, so this time a homozygous black bird, homozygous for the black, is crossed with a homozygous white. And what do we get? We get 100% blue-gray birds, 100% homozygous. So, sometimes the black and the white genes, color genes, can't share and create an in-between. They want to both be seen. So this is called codominance. We see this in coat color genetics. So we have a red cow that has an R gene. We have a white cow that has the W gene. And together, they make an offspring called a roan, which is a mix of red and white hairs. So let's practice this together. A red bull, so big R is red. Big W is white. So these are completely different alleles for red and white. And when you have either the red or the white, you get roan. So this one is asking the offspring from a roan bull and a roan cow. So an RW times an RW are heterozygous. So now we're not going to use homozygous dominant or recessive. You're homozygous red or homozygous white. So we have 25% red, 50% roan, 25% white. Oops, got really far ahead there. Okay. So with codominance, you can be red or white or roan. It works pretty similarly to incomplete dominance. It's just the color that you get is not a perfect mix because they both want to be seen. So now try this one in feather color genetics, where you can be black, white, or erminette, which is the spotted. Okay, so let's check your work. If you've got these, you're black. If you've got this, you're white. And if you've got the black-white, you are erminette. So this is asking the heterozygous phenotype erminette cross 2 erminettes. So that's BW times BW. So black-white times black-white gives us two B's, BW, BW, WW. Okay, so let's see. We've got black. 25% chance of having black, 50% erminette, and 25% white. So now you're going to continue with erminette, and you're going to try this example. Okay, so you can be black, erminette, or white. So this time we are crossing a black, which is two B's, and a white, which is two W's. So two B's times two W's. So black, white, black, white, black, white, black, white. 100% of our offspring are erminette. So there's one last little tricky example of genetics here, and that's called sex-linked traits. So this is where having X, the genes are on the X or Y chromosome, and that influences the ratio that you're going to get, because some of them are much more prevalent in males than females because they only have one copy of the X. So females have two X's, males have an X and a Y. So we're going to start by looking at hemophilia. So we have a normal clotting male, so using X with a capital H is normal. X with a small h is having hemophilia. So it's a small h, so we know it's a recessive gene. So we're taking a normal clotting male, so the male has one X and a Y, and we're crossing it with a heterozygous female. So she has one normal clotting allele and one hemophilia allele. I always put females on the top, but it doesn't matter whatsoever what you do. I just like the way it looks. So let's see what offspring we have. Okay, so they have a 25% chance of having a daughter who's a normal clotter. They have a 25% chance of having a daughter who's a carrier, meaning she's heterozygous but does not have hemophilia. They have a 25% chance of having a son who's a normal clotter, and a 25% chance of having a son with hemophilia. So on these genes where it's recessive, you more often see these appear in the men because they only get one copy of the H. So if their mother is a carrier, they have a 50% chance of having hemophilia themselves. So our last example is in color blindness. So pause the video and see if you can work out the offspring between a colorblind male and a carrier female. Okay, so color blindness is a recessive trait. So if you have X in the big B, you have normal color vision. And if you have X with a little b, you are colorblind. So we have a male who is colorblind, so he's got the XB allele and a Y, times a carrier mother, so she's got the big and the little. So I said I like to put the female on top over here. I think it's just easier to follow it out that way. And let's see what their chances are for their offspring. So we have X big B, X little b, X little b, X little b. We have X big B and a Y, and an X little b and a Y. So they can have a carrier daughter. They can have a colorblind, we'll say CB daughter. They can have a normal son. and a colorblind son. Now I picked this one because my grandmother was colorblind, married a man who was not colorblind, but because she had 2 copies of the colorblind gene, all 6 of my uncles are colorblind. And that's inheritance and variation.